Lecture 19. This is all about solids, and it's kind of a cool little topic present in this chemistry class. It's very much inorganic in all of its scope. So let's take a look at it. And let me refresh your memory by talking about diffraction for a second. You may recall when we talked about quantum mechanics and specifically the Broglie's equation that you basically can have particles moving with some velocity, ultimately interpreted with a wavelength, right? And that's kind of the particle wave duality of matter. And basically, what I explained in that particular lecture was that for a particle that has a wavelength of 0.77, or excuse me, zero point 0.7 angstroms, you end up basically with a particle that travels at a really high speed. We're talking about 10 million meters per second, which is it's a fraction of the speed of light, but it is pretty high nonetheless. And the idea basically that I imparted at that point was that you could have this particle traveling at that speed, and you could do that so long as you have a voltaic plate of opposite potential. So one way to kind of generate the wavelength that you see here, the zero point 0.7 angstroms, is to have an accelerated electron colliding with a voltaic plate made out of some metal target material like molybdenum, in other cases copper. But that collision ultimately, which is given in terms of the momentum of the particle, gets converted to energy in wavelengths, right? And this is where the Boyle's equation kind of comes in. So you can basically generate upon that collision the zero point 0.7 angstroms worth of wavelength, energy wavelength, arising from the 1.05 times 10 to the 7 meters per second speed of the electron. And what I did mention back then was that the wavelength, the X-ray wavelength that you're getting here, is roughly in the vicinity of the size of bond lengths in molecules. So if you have a solid crystalline substance whereby all the molecules are actually arranged in an orderly fashion, you can use that information to get a diffraction pattern that ultimately yields the experimentally determined structure of the molecule. So we're going to deal with this idea of the structure of molecules, specifically in something we call unit cells. Now, the unit cells, you could think of them as being kind of cubic spaces, right? They encompass the x, y, and z coordinates of a system. And the most simplistic of them all is the cubic structure, where all of the sides, right, the length, the width, the height, they're all of the same dimension. Whatever the value is, it will be the same for all three of them. And in addition to that, the angles separating each one of the x, y, and z axes are all 90 degrees, making basically a cube. Now, in the case where you have just a regular cube, where atoms are connected on the corners of the cube, we call that the primitive structure. And since this is a cubic lattice structure, we call this the primitive cubic structure. Now, there's other structures though where you not only have atoms on the corners of the cube, but you also have one in the very center of the cube, and we refer to that as the barycentric cubic structure. In addition, for cubic cells, we also have an additional one known as the face-centered cubic structure, and it's named such because, as you can see, the red atoms right here, represented by spheres, are basically located in the middle of each face of the cube, right? This cube has 6 faces, and in the middle of each one of those faces, you have an atom present, right? And you kind of create this octahedral-like arrangement if you kind of have pseudo connections among them. But the cubic structure is not the only type of unit cell in which atoms can arrange themselves and position themselves. We also have another one known as the hexagonal structure, and the hexagonal structure, excuse me, basically characterizes itself by having one of the angles be equal to 120 degrees. The other two are actually 90 degrees. But this is what gives you this kind of hexagonal shape overall. And if you only have atoms on the corners, and notice here that we're dividing this by 1/3 of the value, right? So if you have atoms in, this rectangular shape out of the hexagonal structure, you basically have a primitive hexagonal structure. And that's for, by and large, the extent of the hexagonal structures. Then you have other ones that are truly rectangular, where all of the angles are 90 degrees, not to be confused with a hexagonal, where one of the angles is 120. But one out of the two sides has a different length associated with it. So notice like here the height is longer than either the length or the width of the actual cube. So we call this a tetragonal structure. And if you have atoms only in the corners, you're dealing with a primitive tetragonal structure. If you have one in the middle, in addition to the corner atoms, we call this the body-centered tetragonal structure. Another one that it's also similar in scope is the orthorhombic structure. Here, not only is one of the sides different than the other two, in fact, none of the sides are equal to each other. So you actually have a true rectangular shape of various lengths, and the lengths don't equal each other. But the angles are all 90 degrees, so you still cannot retain that characteristic. This is known as the orthorhombic unit cell. And once again, have atoms on the corners, you're dealing with the primitive structure. Have one atom in the center, you're dealing with the body center structure. Have atoms on the faces of the unit cell, we call that the face center structure. And the orthorhombic also has one more, in which you only have two faces occupied, opposite faces, and this is called the two-phase structure. All right. Now the next thing is known as the atomic count, and here's where we actually take into account where the atoms are positioned in respect to the unit cell, and we use that information to determine how much of the atom actually belongs to the unit cell and how much of the atom is contributed to the total count in the unit cell itself. And the idea is pretty much this: if the atom is inside the box or inside the unit cell, it doesn't really matter if it's in the middle or off to the side. As long as it's inside the cube, all of it counts as an atom for the unit cell. We call that a center atom. Now the face ones, here the idea is that for it to count as part of the unit cell, it only belongs to that unit cell, period. And the easiest way to determine whether or not all of it belongs to the cell or not is to basically draw additional cubes. So notice here that I drew another cubic structure right on the face atom on the right side. And once I do that, there is no other cubes that I can draw that will encompass that particular red atom in the middle. And what this tells me is that that atom in the face can actually be shared between two adjacent unit cells. So only 1/2 of it belongs to any one of the boxes. So face atoms only contribute half of themselves to the unit cell. Similarly, if we look at the corner atoms, and you can concentrate perhaps on the one right here on the upper right top corner of the cube, you can draw a cube right next to it, and that partly occupies that corner atom. But you could still draw two more. This time I'm going to draw them on top to occupy that atom, but you still have empty space that hasn't been accounted for in regards to the atom. So you could actually draw another 4 boxes right in front of the atom, and at that point you have fully encapsulated the corner atom. But it took a total of 8 boxes to make that happen. And what that means is that the corner atoms only contribute 1/8 of themselves to the unit cell. Okay, finally, there's other atoms which we'll also investigate here, known as interstitial atoms. This actually don't lie on the middle of the face of the cube, but they rather lie on in between the corners and in between the faces, if you want to look at it that way. And for this type of atoms, you can draw another cube on top. So let's concentrate on the one here on the front. And to fully encapsulate it, you need to draw another 2. And when you do that, that atom is fully covered by cubes all around. and he took 4 of them, so these interstitial sites or interstitial atoms only contribute 1/4 of themselves to the unit cell. Alright, so knowing that, we can use that information to basically determine how many atoms we have in a full unit cell. So you could look at the unit cell right here, and the easiest thing will be to kind of subdivide the whole unit cell into smaller ones, but just remember that the entire thing is a unit cell. So here we can identify that there is one center atom right there. Okay, so there's one center atom that contributes all of itself to the unit cell. We also have 6 face center atoms here in red, and each one of them contributes 1/2 to the unit cell. We also can see that there's 8 corner atoms present in the unit cell. Each one of them contributes 1/8 of themselves to the unit cell itself. And then there is a total of 12 interstitial sites. So we have 4 on top, 4 in the middle, and then there's another 4 in the very bottom. So each one of these atoms contributes 1/4 of themselves. If we carry on the multiplications here, we have 1 times 1, which is 1, 1/2 times 6, which is 3, 1/8 times 8, which is 1, and 1/4 times 12, which is 3, meaning that there is a total of 8 atoms present in this unit cell alone. Not to be confused with each individual atom present in all the sites. Here we're counting how many atoms belong to this unit cell only, and only to this unit cell. Alright, now in the next video, I'm going to approach the idea of molecular formulas by counting the atoms present in the unit cell. So hopefully you're kind of beginning to see the trade right here. So remember, center atoms count for 1. Face atoms count for 1/2, corner atoms 1/8, and interstitial atoms count for 1/4 of themselves. Okay, see you in the next video.